Good evening, all. Today I am going to talk about mystifying species, which faces many issues in taxonomy as well as genetics. Muntjacs are taxonomically diverse group, widely distributed in South and Southeast Asia. It is small, solitary deer, which is denoted as a muntjac. The member of the genus Muntiacus are great interest to evolutionary biologists due to its chromosomal rearrangement and ongoing discussion on number of living species in this genus. Members of this genus largely classified based on their morphological characteristics like body size, antler size, their color, coat, and distribution range. And few members were classified based on chromosomal and molecular variation. Currently, more than 16 species and subspecies have been proposed in this genus. However, Group et al. Classified all species in 4 major groups based on their morphological characteristics and its distribution range. Although the classification is not fully accepted because it is difficult to differentiate a species based on limited morphological data when several species of Muntjac share similar morphological characteristics and having overlap distribution range. This is the distribution range of Muntiacus species, which are classified by IUCN. Currently, 13 species of Muntjac are classified by IUCN Red List, which is distributed throughout South and Southeast Asia and southern China. Out of 13 species, 7 species still data deficient across their distribution range. So today I'm going to talk about Indian red muntjac. Red muntjac has widest distribution in compared to other muntjac. It is distributed throughout South Asia, including Sri Lanka, mainland Southeast Asia, and southern China. Red muntjac are the habitat generalists. They occur in very wide range of habitats, from evergreen forest to secondary and highly degraded forest, and from cropland and savanna. It is generally found at altitude of 1500 meter, but few sightings also recorded altitudes 3600 meter. Muntjacs are omnivorous in nature. Apart from plant matter, sometimes it is fed the eggs and young of birds as well as hunt small mammals. Muntjac has independent evolutionary history. Muntjac are so distinct morphological characteristic like presence of small antler, long pedicels, and the presence of permanent upper canine like those of hydropods and mossids. And the canine are enamel coated. Muntjacs are also known as a puzzling deer. They are so similar morphology with large diversity in their chromosome number. The northern red muntjac, which hold lowest number of chromosomes in mammals, reaching at 6 for female and 7 for male, whereas the southern red muntjac have 8 chromosomes in female and 9 for male. Currently, the population of northern red muntjac is decreasing across their distribution ranges. The red muntjac largely face threat from hunting for meat, skin, trophy. They are being used in traditional medicine across distribution range. The high anthropogenic pressure, which result in fragmentation of suitable habitat, also affect the red muntjac population across their distribution range. The northern red muntjac is currently listed as least concern in IUCN Red List and protected under Schedule 3 for of the Indian Wildlife Protection Act 1972. These are some trophies of red muntjac. Red muntjac has one of the most controversial discussed taxonomies. However, over the period, multiple taxonomic classifications have been proposed. Presently, based on the chromosomal variation, two species of red muntjac classified by IUCN, the northern red muntjac, Muntiacus vaginalis, and the southern red muntjac, Muntiacus muntjac. The Indian red muntjac distributed India, Sri Lanka, Nepal to Bangladesh, Bhutan, and mainland Southeast Asia and southern China. Whereas the southern red muntjac distributed mainly Malay Peninsula, Sundaland, Sumatra, Java, Bali, and Borneo Islands. Over the period, multiple taxonomic classification of red muntjac has been given by scientists. First, Macaulay et al. Classified red muntjac as a single species, Muntiacus muntjac, and 10 subspecies in this group. Grubb and Groups et al. Recognize 6 species along with 2 subspecies of Muntiacus using geographical distribution and morphological characteristics. IUCN currently recognizes the classification of red muntjac given by Groups and Grubb in 2003. Timmins et al. Validate 2 species of red muntjac across their distribution based on their chromosomal variation, which are Muntiacus muntjac, known as the southern red muntjac, and Muntiacus vaginalis, which is known as a Indian red muntjac. Recently, the complete mitogenome sequence indicates the presence of 3 distinct maternal lineages across the distribution range of red muntjac. Sri Lankan red muntjac from Western Ghat, India and Sri Lanka, Northern red muntjac in northern India and Indochina, and Southern red muntjac found in Sundaland and other islands of Southeast Asia. Based on above taxonomic complexities, we further investigate the phylogenetic relationship of red muntjac across the India to validate their phylogenetic relationship. We generate 20 whole mitogenome of red muntjac. The sample collected from Western Ghat, India, northwestern part, central India and northeastern part of India. We also compare our mitogenomic data with previously published data by Martins et al. Our phylogenetic result showed that all sequence of red muntjac cluster into 4 major clades. Clade 1 consists the mainland red muntjac, M. vaginalis. Clade 2 comprises the individual from the northwestern part of India, that is from Uttarakhand, Punjab and Himachal Pradesh. Clade 3, in clade 3 the sequence mainly from Sundaland, Muntiacus muntjac. All sequences are from Sumatra, Malay Peninsula, Borneo, Java, Bali Islands. Clade 4 consisted of individuals from Western Ghat of southern India and Sri Lanka. We also calculate the genetic distance based on whole mitogenomic data and compare with other Muntiacus species. We found the M. aureus, which is distributed in northwestern part of India, genetically 2% different from M. vaginalis, and however the Malabaricus is more distance in among red muntjac, showing more genetic distance among red muntjac group. Malabaricus showed around 3.9% genetic distance with M. vaginalis and 4% genetically distance with M. aureus. Coming to conclusion, our finding validate the presence of Himalayan red muntjac within the red muntjac group from northwestern India. Primarily, this study resolved the phylogeny of red muntjac and delimited 4 distinct clades, namely M. aureus, M. vaginalis, M. malabaricus, and M. muntjac. The metagenomic data set provided will play a vital role to study the evolution of genus Muntiacus. The output of study will directly help conservation authority to prepare effective future management plan along with strategical planning of in situ and ex situ conservation of different lineages in India. Thank you.